Hello friends, is Botany next episode. In this episode, we'll be discussing about cell cycle chapter, second part. So we can proceed. So I will share the screen so it will be convenient for you. I think a screen came, so we can proceed. So what all topics we'll be discussing in this episode? Chapter is chapter 10, cell cycle and cell division, second part. So one introduction, then mitosis, then amitosis, then as usual, 5 theory question, 5 NEET model question with the key. So when we start to enter the topic as an introduction, in animals, mitotic cell division is seen only in diploid somatic cells. But in plants, mitosis is seen in both haploid and diploid cells. Mitosis is also called as equational division because two daughter cells will be formed. Will have same number of chromosomes like that of parent cell, identical. Mitosis is also called as somatic cell division because soma means body. So this cell division normally occurs in body cells. In plants, it occurs mainly in growing areas. In animals, that is seen in body cells. Gonads also we can find this mitosis, but we cannot believe it. In gonads, where it is seen? In gonads, it is seen in undifferentiated germ cells. And mitosis is involved in repair mechanism. It will replace old and worn out cells. In man, daily around 5 into 10 raised to cells will be lost from skin, alimentary canal, lining, WBC, RBC, etc. And the lost cells are replaced by mitosis. So 5 into 10 raised to 9 cells all will be replaced by mitosis. So that is an introduction about this cell division. Now we can proceed to mitosis. See the root word mitos, it is Greek, means thread or fibril. And first observe what we believe is Strasburger in 1870 in plant cells, in 1879 Bovary and Fleming in animal cell. In 1882, term mitosis was given by Fleming. The term mitosis came from Greek, mitos means thread. So many scientists are there, some famous scientists just mentioned. M-phase, that is often called mitotic phase, or we will be normally calling it as most dramatic phase of cell cycle, divided into two. One, karyokinesis, two, cytokinesis. Which item we have to proceed? So first we can proceed with the karyokinesis. Again, carry-on Greek word means nucleus. Kinesis means movement, energy, transport, etc. So for convenience, mitosis is divided into 4. Originally, there is no separation like this. For convenience only, we made it. 1 prophase, 2 metaphase, 3 anaphase, and 4 is telophase. And this diagram is familiar to you. It's a typical NCERT famous theory entrance question showing all events in a cell cycle. Hoping that first page is clear to all of you, we can proceed to second page. So among this karyokinesis, first phase is prophase. First stage in karyokinesis, it is after G1S, G2, can be divided into 3. Early prophase, mid prophase, late prophase. Now among them, first is early prophase. So when we discuss early prophase, shape of the nucleus becomes spherical. Cytoplasm viscosity increases. And DNA, which was existing like a chromatin reticulum, we cannot identify clear chromosome in network. Here what happens is DNA forms elongated chromosome. Thus chromatin reticulum disappears. And this overlapping of elongated chromosome we can observe. Appearance of chromosome like a ball is called, that stage is called spireme stage. And that is seen in early prophase. In animal cell, centriole pairs start to move towards the opposite side, animal side. Higher plants, no centriole. So in animal cells, centriole pairs start to move towards the opposite side. That is towards poles. From center move towards the poles. So centriole have some radiating fibers or fibrils, which is called astral rays. Centriole with the astral rays together called asters. This diagram you can find here. That is connected with Anaphase, but all these items it is marked there is previous NCERT diagram. Presently you cannot observe it, but everything we can observe here. Everything. That's why I put the diagram. So asters, and in animal cell you can find two asters, and they're connected by a spindle. So two asters are there, so it is called amphiaster. In plants, asters will be generally absent and astral. Aster means a star. That is how the term came. Animal side it is amphiaster, plant side it is an aster. Now we can proceed to mid-prophase. So in the prophase beginning, even distribution of chromosome in the nucleus. As the stage proceeds, mid-prophase when we reach, they move towards periphery. As a result, a clear area can be seen at the center. See, something like this way. This is anaphase still. Center. Chromosome reduces its size, getting thicker. And two chromatids. This is the diagram, chromatids. We will be studying it. Structure of chromosome in cell, the basic unit of life. So two chromatids present, which are otherwise called sister chromatids. So within a chromosome, chromatids are called sister chromatids, and joining part of the chromatids are called centromere. This red mark you can find, centromere. I think up to that it is clear. This is a diagram, very, very meaningful and sensible. Now comes late prophase. In late prophase, nucleolus and nucleoli disorganize. Golgi body, endoplasmic reticulum also not seen. Amphiastral spindle. It is called centric spindle, while anastral spindle is also called as eccentric spindle. Because that two term not in NCERT but previously came. Centric spindle, amphiastral spindle, while anastral spindle is called eccentric spindle. Spindle apparatus has got maximum diameter in the middle, and main ingredient of the spindle is tubulin protein. I think this page is clear. Late prophase is important for exam. Nucleolus, nucleoli, disorganized, Golgi body, endoplasmic reticulum also not seen. Spindle fiber is made up of tubulin protein. I think this page is clear. We can proceed to next page. Next is metaphase. Meta's got different meanings, which include not first. What means second. Otherwise, after. Metaphase begins with the disintegration of the nuclear membrane, that is end of the prophase. We can always call prometaphase. Thus, chromosomes spread in the cytoplasm. Chromosome become thickest and shortest. Thus, morphology, number of chromosomes can be studied in metaphase. Morphology. Completion of mitotic spindle formation. Now, in connected with metaphase, there is a famous term, congregation. So, what is meant by the term congregation? Congregation means process of bringing the chromosome from the periphery to the equator towards center is called congregation, that is connected with the metaphase. And then we can find centromere of all the chromosome forms an apparent plate called metaphasic or equational or equatorial plate. Metaphasic or equatorial plate. One more point, exam point of view, colchicine, a mitotic poison, works at metaphase stage, prevents spindle formation, by which prevents separation of chromosome during the anaphase stage. Think idea clear? Now we can proceed to third area, anaphase. Ana Greek means move or up or even separate likewise. Centromere, in this diagram you can find it, this one. Centromere of each chromosome divides into two. That is why two chromatids we got. Here it gets splitted. Now each chromatid, after separation of these two, will be calling it as chromatids. So each chromatids have its own centromere, later become 2 daughter chromosomes. They move towards respective poles. See to which place they are moving from center to periphery. Respective poles of the spindle. And connected with anaphasic movement, many theories are there and many fibrils are involved in it. And centromere leads the path. Centromere shows the direction while limbs follow it. And in anaphase, we can find shape of the chromosome can be best studied. Anaphase. Metaphase morphology. Here it is anaphase. Chromosome shape. And 4 shapes you can find V, L, J, I. V is otherwise called metacentric, L is otherwise called submetacentric, J is otherwise called acrocentric, I is otherwise called telocentric. And at the end of anaphase, we can find 2 groups of chromosomes are formed. Now comes fourth one, telophase. Telos, Greek again, means end. So in telophase, what we can find just opposite to the event seen in prophase, because that is starting stage, this is ending stage. Cytoplasmic viscosity decreases. Chromosomes again changing into chromatin. Mitotic spindle disappears because its function over. Nuclear envelope develops. That all disappear during end of prophase. Nucleolus, endoplasmic reticulum, Golgi body, and all other organelles reappear. That is telophase. Now comes, after karyokinesis, cytokinesis. Kaitos means empty or hollow. Kinesis, again movement. Here cytoplasm divides into 2. This stage denotes end of the cell division. In this cell division mitosis, nucleus and cytoplasm divides. If karyokinesis is not followed by cytokinesis, karyokinesis not followed by cytokinesis, results in multinucleate condition, cell with more than 1 nucleus. It is otherwise called syncytium or syncytic condition. It is due to lack of cytokinesis after karyokinesis. Now, when we come to cytokinesis, a comparison we can make between plant cell and animal cell. It is different. Now, first is plant cell. It is called cell plate method. While in animal cell, it is called cleavage furrow method. See the cleaving furrow. This is plant cell. This is animal cell. And in plant cell, it is cell plate that is from center to periphery, moving towards periphery. It is called centrifugal cell plate in plant cell. Well, animal side, it is from periphery to center. It is called centripetal. It is by cleavage furrow. So plant side, it is centrifugal cell plate. Animal side, it is centripetal. It is cleavage furrow. I think that idea is clear. Now, plant cell plate. How that plate formation takes place in connection with an item called phragmoplast that is formed, which forms cell plate later. Now, importance of mitosis. Mitosis involved in maintaining of size, shape, nuclear cytoplasmic ratio, wound healing, regeneration, chromosome number maintenance, DNA maintenance, connected with reproduction, surface area volume ratio, etc. So a large number of functions it has to do. Now a variation from mitosis, it is called amitosis, which is otherwise called direct cell division. It is a mode of reproduction, asexual. So originally asexual cell division, where no chromosome, spindles, etc. are not involved. So no chromosome, no spindles, nothing will be there. It is more commonly seen in acellular organisms like bacteria and protozoa. Again, areas like old cell, disease cell, fetal membrane, cartilage cell, etc. It was discovered by a famous scientist, Raman. And what happens here? The diagram I will show you. Initially, nucleus of the cell elongates. See, elongates. After that, a constriction appears in the nucleus. Here you can find nucleus gradually get deepens. Divides the nucleus into 2 daughter nuclei. Similarly, constriction appears in the cytoplasm, which divides the cytoplasm and nuclei into 2 daughter cells. Here, no spindle formation, no distinct chromosome, nothing will be there normally. Again, nuclear envelope remains intact. Size of the cell formed may vary. Some area, some books reference say it will be almost half, double, etc. So may vary. And that is regarding this famous mitosis. And the questions, 5 theory question, 5 NEET model question, and one more thing. Here we can find the diagrams of mitosis. NCERT, so diagram-wise question. First is early prophase. When we come to late prophase, you can find these membranes are disintegrating. And when we come to early metaphase, chromosomes are coming towards center. And what we call that name congregation, and here it is center metaphasic plate. Then chromatids of the chromosome separate in anaphase. Then it is converting into 2 cells. That is regarding diagram. So what all things we learned in this episode? So basically, we learned 3 items with a set of questions. Introduction, mitosis, and mitosis, where the mitosis will take place. Why it is called somatic cell division. And we can also find plants haploid and diploid. Mitosis will take place. Animal side only diploid somatic cell. Growing area takes place. And in gonads, it is an undifferentiated germ cell replacing. Now, when we discuss the function, replacing the damaged cells. Similarly, old cells, etc. Then lining, elementary canal lining, WB service, all this will be replaced by lost cells will be replaced by mitosis. Mitosis. Now, meaning, Greek meaning, scientist, observed, plant cell, animal cell, mitosis, dramatic phase, 2 division, karyokinesis, cytokinesis. Karyokinesis, again, meaning of the term, 4 stages. After that, first we entered into prophase. After G1 as G2, early prophase, mid prophase, late prophase. Then after that, we entered into metaphase, metaphasic plate, thickest and shortest, chromosome morphology. Then we entered into one more item, that is colchicine, preventing spindle fiber formation, mitotic poison, effect comes in metaphase. Then anaphase, separation. Then 4 shapes of the chromosome. Shapes of the chromosome we can study in anaphase, VLJI, metacentric, submetacentric, acrocentric, telocentric. Then comes telophase last, opposite to prophase, antagonistic phase of cell cycle, telophase and prophase. After that, cytokinesis. Cytokinesis 2 types, plant side and animal side. Plant side it is centrifugal by cell plate. Animal side it is called centripetal with a cleavage furrow. Then again, importance of mitosis. Then amitosis, direct cell division, asexual, no chromosome, no spindle, seen in acellular organism commonly. Otherwise, some other area, etc. It is in scientist stomach. And what happens? 3-step procedure. Here we can find nucleus elongates, constriction appears, then get deepens, divides nucleus into 2 daughter nuclei. Similarly, constriction appears in the cytoplasm, which also divides. Finally, we'll be getting 2 cells. And figures I shown here. NCERT figure, important theory and runs. Then after that, 5 theory question, 5 NEET model question. So this episode ends here. If you like this episode, like it, share it, subscribe all, comment on it. We can meet later. Take care. Thank you.